Hey everyone, welcome to the Bio Breakthroughs podcast. I'm your host, Jared Taylor. Joining me today is the CEO of Biological Dynamics, Paul Billings. How are you today?
Great. Thanks for having me.
I'm excited to have you. Let's dive right in. Give us an overview of your background, and then we'll talk about the company.
So I'm a physician. I did a MD and PhD many years ago at Harvard Medical School, working with Nobel laureate Baruch Ben-Asaraf on some questions of T-cell biology. I then trained as an internal medicine doctor and as a medical geneticist, and have spent about half my career in academic settings, both at Harvard and at Stanford and in UCSF. And then the second half of my career has been largely in the biotech and commercial sector. I've been the CEO of a couple of companies, most recently Biological Dynamics, which we'll talk about a little bit more in a minute. But I've also been the chief medical officer of both public and private companies, and have had the opportunity to really see the development of personalized and then precision personalized medicine as a new tool to help treat people. And I was a practicing physician for a good part of my career. So I was actually able to see it work in the clinic, which is even more gratifying. So that's me. Right now I'm a CEO and a director of several companies, and really interested in seeing the new things happen, making medicine better.
And thank you so much for that great overview, by the way. Talk us about Biological
So
Dynamics
Biological
today.
Dynamics is more than 10 years old, and has spent the vast majority of its time and money over the last 10 years developing a really breakthrough technology, which allows, in a very small amount of blood, less than a half a cc of blood, the ability to isolate particles that are present in the plasma, in the liquid part of blood. And in that, with that technology and with all that development, we've been able to focus on some particles that are present in the blood, which were largely unexamined historically. We historically we looked at proteins, we looked at the cells in the blood, those kinds of components. These particles called exosomes are incredibly abundant and were largely unexamined. But we've been able to create a rapid and effective and cheap way for people to examine it, and then we've of course examined it as well. And we found that it's a really that exosomes are abundant, and that their cargo, the things that are inside these little vesicles that we isolate, are very informative about the cells that produce them. And so we've turned that observation onto questions which have plagued physicians and patients since the beginning, and hopefully are beginning to produce some really interesting new answers and solving really difficult problems in healthcare.
Thanks so much. Yeah, it's I always ask, right, because you have been around for 10 plus years, it's interesting to hear where things are at today through your lens. And you're solving some really great problems in healthcare. Talk us through, how you see your technology today helping with many of the problems that you solve, where it's heading in the future.
We've just really stabilized and commercialized the technology, and we've just actually begun to accumulate the clinical evidence that we need to show how the technology will benefit doctors and patients, right? So we're in I would say, the rapid data clinical, practical data accumulation phase, as well as distributing our instrument and our consumables to researchers all over the world, so that they can use the instrument and develop corroborative data, new insights, using the isolation of exosomes as a tool to help them. And then, of course, we learn from their work as well. So we're right in this transitional phase, and I've seen it at other companies as well that I've been at over my career, where we've got a new insight, The inside is reliable. We've put it in, we can do it at clinical grade. And now we're going to accumulate a whole bunch very quickly of clinical evidence about applications, about ways that it provides insight. And that's where we are now. We're most advanced in pancreatic cancer and in the early detection of pancreatic cancer. Pancreatic cancer is one of the most deadly cancers that are out there. It's probably now the second most deadly cancer after lung cancer in terms of the number of cases per year. And pancreatic cancer is one of those cancers where if you don't diagnose it and treat it effectively in the very earliest stages, your chances of survival are very, very much diminished. The 10-year survival or the 5-year survival for pancreatic cancer is on the order of 10%. But if you catch it at stage 0, which is the very earliest stage, or stage 1, where you have a very, very little tumor in the pancreas, the 5-year survival is at least 50% and probably higher. So just shifting the time of diagnosis, the stage of at the stage of diagnosis, gives you a 5-fold increase in 5-year survival. That's an amazing statistic. So we're all about identifying people who should have regular detection, regular surveillance of their pancreatic cancer risk, and then providing them with the most effective surveillance, better than imaging, better than other blood tests, the best in class, and then getting more and more people diagnosed early and treated effectively. That's our first target. We'll have others, and we're going to stay with that theme. We're going to stay with themes where there are people at risk. If they get caught early, they're going to be treated and they're going to be cured. If they're caught late, they're not going to be cured with the treatments usually. And we're going to provide the best in class surveillance for those patients.
Now, in your space, what are some of the key ways that you feel you differentiate from those other
So
and moving forward?
we're largely in the so-called liquid biopsy space, and the liquid biopsy space is a space in which people are trying to use blood tests, mostly, sometimes urine, sometimes saliva, but mostly blood tests to replace what used to be very invasive surgical or needles biopsies. Right? We used to absolutely need tissue to make a final diagnosis of a disease, to make the prognosis and to set the treatment plan for that disease. And largely we still need tissue at some point along the diagnostic and illness odyssey to better inform care. But the more and more we can shift it to blood testing or to other kinds of fluid testing, the better it is for the patient. The more often you can do the procedure, the surveillance, for instance, in our case. And so it's a better situation if you can do it in the blood. We've been trying to do it in the blood for decades. We had protein tests, we have simple blood tests, we have more complicated esoteric blood tests. Most recently in the liquid biopsy space, we've had the use of the analysis of DNA in the blood, particularly DNA produced by tumors. If your disorder that you're studying is a cancer, and looking for circulating tumor DNA and then analyzing it in various ways, whether you analyze it for targets to treat or you analyze simply the amount to decide whether there's a tumor producing a lot of circulating tumor DNA or not. Anyway, DNA, some people use RNA. We use exosomes. And the reason we like exosomes is that they're incredibly abundant and they're produced all the time, no matter what. We've not found a situation where there's a normal cell or a cancer cell, for that matter, that isn't producing these vesicles during their entire life. Circulating tumor DNA, for instance, is produced mostly by the death of cells. And that death of cells can be the normal death of cells, or can be done because there's an immune response to those cells. But the bottom line is it requires a death, and that's a disordered cell, and there are limitations to what kinds of cells will produce cell-free DNA, for instance, and put that material into the blood. Those limitations don't seem to be true for exosomes. So we're focused on exosomes and on using exosomes at all stages of the diagnostic odyssey.
And you know, when you're improving outcomes, how how would you say the platform and test is currently being used by?
Okay, so it's very early on, as I said. So we're in the we're just learning how physicians and patients best use our technology, and there may be other applications of it that are that we just haven't yet fully gotten a data set for, but may be very important. But right now, the way physicians are using it is identifying a high risk patient, let's say for pancreatic cancer, and that people might have a genetic reason, a family history reason, a fact of their past clinical event that, let's say they had pancreatitis most of their lives, or they have a cyst, for instance, in their pancreas, and a lot of people have cysts in their pancreas. And the question is, we are asking physicians to use our test along with the current guideline recommendations, which are mostly about imaging and biopsy, and use our test to decide whether that's clinical situation has converted to an early stage cancer or is still benign. If it's still benign, they can wait for another 3 to 6 to 9 months and do the procedures again. Or if our test says that it's not benign anymore, then they need to be more aggressive in further working up that lesion that there's concern about, and presumably removing it as quickly as possible, because we know that without removing it, those lesions progress and reduce life expectancy.
And again, you said things are still early, but what are the next steps? What is some of the part that really excite
Well,
you about what's
applying it
next?
in all sorts of populations. I already told you, patients with pancreatitis, patients with cysts, patients with a family history or genetic findings that put them at high risk for pancreatic cancer. Those are and so we're participating in clinical trials that are using patient cohorts from all over the world to really further identify exactly when we can and what frequency patients should have the surveillance. Obviously, pancreatic cancer is a disorder mostly of people above the age of 50. If you're at risk but younger, do you need to have the surveillance as frequently as someone above the age of 50? Well, probably not, but we really need to have firm evidence of all that. So we're answering all sorts of really applied, relevant questions of how to use a very highly sensitive and very effective surveillance tool in a particular deadly disease. Now, we're going to take lessons from that, and then we're going to turn that information on ovarian cancer, for instance, and on lung cancer, and on other really deadly cancers where, while we're making important treatment progress, still early detection and surgical removal is still the mainstay of treatment approaches. And that's where my company is going to make most of its efforts. Now, there are other, we may create with that information, with that experience, other products. For instance, we might create a product which you could use not even in high risk people, in average risk people across a lot of cancers. And there are other companies, of course, and other investigators who are trying to do that right off the bat. We're starting with high risk, high value patients and trying to make, save some lives very quickly. But we're learning, and we may apply it to other cancers. Our technology works in other disorders. There's been a lot of excitement around new treatments for Alzheimer's dementia recently. Dementias produce exosomes that are detectable in the blood. So we're applying our technology in very early trials to see whether we can identify different kinds of dementias, including Alzheimer's dementia and traumas to the brain, for instance. And that may turn out to be another area where we can apply our knowledge to the benefit of patients.
Well, I'm excited to stay up to date with you and hear where things continue to progress. And hopefully, we can have you
I'd
come
love
back on
to.
again in the near future and dive more
Thanks for
into
having me.